How do you think will iCLIP now resolve the mechanism of RNA-related diseases?
Right, so this is actually what we are most excited about right now, and I think the field as a whole. There's a real opportunity now with developments in genomics, imaging, structural biology, and so forth. And the unique aspect of proteins that bind to nucleic acids is that they are very rich in regions that are disordered in solution. So in addition to these defined structure domains, they also have sequences that are very difficult to study with classical biochemical approaches. And these are actually the sequences that tend to accumulate mutations leading to disease. It's very difficult to actually understand the mechanisms of these mutations one by one. And by taking this in vivo approach, analyzing the mutant proteins in an in vivo environment and then monitoring the changes in the assembly of this protein RNA and protein-protein complexes with more high-throughput methods, we can gain the first insight and really direct our further research to that.
What future questions would you like to answer?
Right. So we have now very specific hypotheses that have been derived from the analysis of iCLIP data. And on one hand, we need to test the same hypothesis in more disease-relevant cell models. So at the moment, we have done most of our work in standard cell lines, but we work with collaborators who have cell lines that can produce specific type of motor neurons or glial cells and see how these complexes are actually interacting with RNA in very specific, controlled, cell type specific manner. And at the same time, we also want to combine the information from the iCLIP with imaging and other biochemical methods to really understand what is the sequential mechanism in how mutations cause disease.
You're also working on TDP-43 assemblies. So what are the challenges you're currently faced with?
Yes, so we have been, TDP-43 is a protein that is sort of implicated in ALS, amyotrophic lateral sclerosis, and this tends to aggregate in these patients and therefore then leads to motoric deficits and so on that is known from these neurodegenerative diseases. What we have done, basically, we have done a study to investigate in vitro the behavior of this protein to first make it basically suitable for high resolution analysis, where we're still facing challenges, but we've established a system where we can now study this in the test tube. Basically, we identify the regions that are responsible for this kind of abnormal aggregation as it occurs. And now this is sort of a starting point where we can dive further into identifying and also investigating these regions that are responsible for the abnormal aggregation more in detail.
What are the big challenges studying this long non-coding RNAs?
For many of them, actually. So the thing is, long non-coding RNA, they are classified as one large categories, but probably they act through many different processes and mechanism of action. So I think it's the big issue is to know what they do and how they do it. So we start to have some answer, but still many things to do in that direction.
Now that we are facing this incredible huge amount of new transcripts, which have apparently no coding functions. I say apparently because one has to be very careful before defining that an RNA is really non-coding, because now many short operating frame can be found in non-coding RNA, so that's another issue to be solved. But I think that the effort that we are putting on this system really will help to dissect new mechanism of gene expression control, so provide also new conceptual categories for understanding how our gene works and how this regulation is assists many other factors, transcriptional factors in controlling important processes like cell differentiation.
The most recent leap forward in the exosome field?
I think there are actually several. One is not related to what we do in the lab, is the discovery by systems, more global systems approaches of the substrates, the range of substrates the exosome targets, and the adaptor molecules that come in. So this is one aspect. The other aspect is the interplay of the exosome with other big molecular machines, particular the ribosome. So there are connections with the nuclear ribosomes, pre-ribosomes. There are probably connections with the cytoplasmic ribosomes. And, there is probably much more to come about this. The exosome has been shown to be somehow at the site of transcription, to take care of the cryptic unstable transcripts that polymerase II makes. So, I can imagine in the future there will be connection with the polymerase. There might be connections with the proteasome. So I think, at the moment we have understood the core particle. We are advancing on understanding the regulators and how the regulators interact with the core particle to make it function. And I think the next level is to understand how the core particles, the regulators and adapters interact with different machines in the cell. And so I think that's going to be very exciting. I think the future to see, the exosome working on the ribosome, or potentially the exosome working on the polymerase together with the polymerase. I think, and it goes hand in hand with developments in EM, in technology. So I think it's a great time.
What do you think is kind of like the future of NMD? Where are we moving towards? How are we trying to comprehensively understand the NMD pathway?
Well, the future is really exactly to understand, because we are still, despite of 30 years of NMD research, we're kind of lacking kind of a mechanistic understanding. So that will be key. Understanding really, as I said, what distinguishes whether NMD is elicited or not. And then, of course, I think it has a strong medical aspect as well, because there is at least 10%, some estimates go higher than that, of all known disease-causing or disease-associated mutations that actually result in an RNA with a premature termination codon, which then would be presumably a substrate for NMD. And so it means NMD is an important modulator for the outcome or the clinical manifestations of such mutations. And I think there is still lots to be understood, and then also the understanding will hopefully lead or give hints to very specific therapeutic approaches that can be developed.
And it would be nice just to get your opinion finally about what you think the biggest questions are remaining about the biogenesis or the functions of microRNAs in the field.
I think like in every aspect of, almost in every area, for me at least the important thing is regulation, how these events are regulated in a tissue-specific manner, how they are regulated during development, and because in the end, in the case of microRNA, what is important is to maintain a tight control because it's a very amplifying effect. If you imagine, it doesn't seem like a lot, but you have one microRNA, but it goes from level 1 to level 10. The problem that this microRNA, now we have a lot more, this regulates hundreds if not thousands of genes. So the effect can be, so I think the remaining question, well, you need to know a lot of mechanistic detail, but I think the main question is regulation.
So why do you think looking at RNA biology is so important in trypanosomes?
Well, trypanosomes have this process called RNA editing in the mitochondrion, and none of that is in the mammalian host. Mm-hmm. So if that process is disrupted one way or another, then it would be the magic bullet that could remove both leishmania, the causative agents of Chagas disease, sleeping sickness, pretty much all at once. So and also, of course, it's very interesting from the perspective of molecular biology, how this all operates. Mm-hmm. So that's why we are studying.
Where do you see your lab going in the next few years, and what are the contribution you think your lab will give to the field?
We have one, if I may just bring one. Mm-hmm. So there is, so everybody knows trypanosomes, or at least many people. But trypanosomes are not alone. There is two groups next to them, and one is euglenids. So flagellates that freely live in water. And the third group is called diplonemids. And nobody knew diplonemids. They were considered obscure, non-important. Few aficionados knew they ever existed. But now we participated in a study of the world ocean, where thousands and thousands of samples were sequenced, everything what is there, and we actually helped to discover that diplonemids are extremely abundant in the ocean, and nobody knew it, which is surprising, because you have now environmental PCR, why would you miss a big group? But that still happened, and nobody knows. So we have now probably somewhere around 10,000 species, and we don't know how they look like, not even a single one. So we don't know whether they are parasitic or free living. We know that they have very special mechanisms in their mitochondrion, because one species has been studied and can be cultivated. But again, it was studied by one lab in the world because it was just a bizarre protist. But now we know that they contribute to the well-being of the ocean tremendously. They may be the third most diverse group of eukaryotes in the world ocean.